Hi class, it's Dr. Linder. In this short video, we're going to look at the beginnings of the cardiovascular system. We're going to be starting with the review of the blood. So if you think back to blood, blood is connective tissue. It's liquid connective tissue. And one of the things that differentiates epithelial tissue from connective tissue is epithelium is a bunch of cells closely packed together with little extracellular matrix. But here in this picture, we see a bunch of cells scattered far apart from one another with lots of extracellular matrix. So the ECM, or the extracellular matrix, is that greenish yellow that's surrounding all the red blood cells and white blood cells. That's called plasma. In blood, the plasma and the extracellular matrix portion, that makes up 55% of the blood, whereas the formed elements, which we're looking here at the red blood cells, white blood cells, and platelets, that makes up about 45% of the blood volume. So, all right, let's take a look and just review some of them on this blood board, this plaque here. So the erythrocytes are the red blood cells. Those are these. Let's look at the platelets, get them out of the way. That's over here. Those are platelets. They're involved in forming clots. Let's look at the granular basophils, eosinophils, and neutrophils. So let's look at the granular white blood cells, which are basophils, eosinophils, and neutrophils. Right around the 7 o'clock position, right here is the basophil. Those have dark stained granules. On a microscope, typically the nucleus is somewhat obscured. If you go and look at my playlist on blood, you'll be able to see the microscope slide of the basophil. And you see the nucleus is kind of obscured by the basic or alkalinic staining granules. The basophils typically are involved in allergic type of reactions. They degranulate, they do release some histamine, as do mast cells. Mast cells and basophils are kind of similar in function. Let's look at the eosinophil next. So there's the basophil. Let's look at the eosinophil right in the center. The nucleus is bilobed. 2 heads to the nucleus with larger red stained granules. That's an eosinophil. Also, when high, you're thinking an allergic type of reaction or a parasite. These release histamine ACE, which is an enzyme that tries to degrade and break down some of the histamine that the basophils and mast cells will release. Mast cells we do not see here. Let's look at the neutrophil. So that's basophil, eosinophil, also granular. The neutrophil is the last one that's granular. And just below it, or slightly to the right of it, these are neutrophils. Neutrophils have 3 to 5 heads to the nucleus. So this is 1, 2, 3. This is 1, 2, 3, 4, 5 heads. Neutrophils are your primary responder to bacterial infections. If you have a bacterial infection, neutrophil count will be high. All right, so we did the 3 granular sites. We did the erythrocyte first, the red blood cell. We did the plasma, and we did the platelets. We did the 3 granular sites, the basophil, eosin, and neutrophil. Now let's look at the 2 agranular sites. Those are lymphocytes and monocytes. So if we look down somewhere around between 6 and 7 o'clock right here, this is a lymphocyte. Notice how big the nucleus is, almost as big as the cell. Same thing here between the 12 and 1 o'clock position. Again, the nucleus is just as large as the cell. Same thing here with this lymphocyte. Very different than a monocyte, on the other hand. If you look at the monocyte, it's the largest cell, and the nucleus is kidney bean shaped. Monocytes are what become macrophages when they leave blood vessels and they dive into the tissues. They go from a monocyte to a macrophage. If it's a macrophage in the neural system up in the brain, we call it a microglial cell. If it's in the liver, it's a Kupffer cell. If it's degrading oxidized LDL, they're called foam cells. If it's in the lungs, they're called alveolar dust cells. So macrophage just changed their names, okay? Let's take a look at them individually. So here is a neutrophil. Neutrophils have 3 heads to the nucleus. Their granules are much, much smaller than those of an eosinophil. Eosinophils are bilobed, 2 heads to the nucleus, larger, clearly stained granules. This is taking up an acidic type of stain. And here's our basic granules. These take up a basic stain. Typically under a microscope, the nucleus here would be somewhat obscured and difficult to see due to the dark nucleus plus the dark granulations covering the nucleus. So this is just the model, okay, but that's a basophil. Here's our lymphocyte, where the nucleus is pretty much as large as the cell itself. It occupies that same amount of space. When you think of lymphocytes, we think of B lymphocytes and T lymphocytes. And finally, our last agranular site is a monocyte. So the lymphocyte is agranular and the monocyte is agranular. It's a large, large cell. The nucleus is kidney bean shaped. These are not granulations. These are just, looks like some of the paint chips may have come off of the cytoplasm here. So that's the blood. Now blood travels through blood vessels. So let's take a look at this model. This model is the blood vessel model, okay? Right in the center, you'll see that we have red, and then we have blue. The red one is the artery, the blue one is the vein, and off to the right is just the vein cross-section, so we could see some of the layers. Now, let's get back here. So I think of 3 main layers when I think of a blood vessel. I think of a tunica interna, a tunica media, and a tunica externa. Those are the three main layers. Tunica interna, the internal layer, is sometimes referred to as the tunica intima, or the endothelial, which is made up of simple squamous cells. Then we have the tunica media, which is the tunica musculosa. It's smooth muscle. That's what makes the blood vessel constrict or dilate. And then we have the tunica externa, which is more elastic. Okay, so let's take a look right here at the artery. Let's look at the internal layer first. We see the arrows pointing to the endothelium, which is simple squamous epithelium. It's 1 cell layer, so these are easily damaged. This is why on blood work, we always want to look for homocysteine levels, and I equate homocysteine, you've heard me say it before, as liquid sandpaper, which can damage blood vessels. And when that happens, when this 1 cell layer becomes damaged, the liver produces a lot of cholesterol to try and patch this up. When cholesterol accumulates, it doesn't build up in the lumen and block the lumen, it dives deeper into between the endothelium and the smooth muscle layer, okay? And the macrophages leave the lumen of the blood vessel, go into that other tissue and engulf it like phagocytically. Now they're called foam cells, and those foam cells can accumulate in the deeper layer. And when they start to accumulate, they're called lipid pools. And the lipid pool can accumulate and accumulate and accumulate here, and it starts to bulge into the lumen, and it obstructs the luminar flow where the blood flow is traveling, and it becomes very turbulent. If the blood flow becomes turbulent, the innate intelligence of the body thinks you got shot or stabbed. So that's when the body starts to lay down fibrin, and it clots, and that feedback loop kicks in. Positive feedback loop. It keeps on laying down fibrin until a clot is formed. And if it's going up to the brain, it's a stroke. And if it's to the heart, it's a heart attack. So there's a little clinical application there. So here's the tunica intima or tunica interna, which is made up of simple squamous epithelium. Sometimes it can be referred to as the endothelium. So many terms for that. As we move externally, the next important layer that I want you to know is the tunica media. The tunica media is in the artery and in the vein, as is the tunica intima or interna. It's in the artery and the vein. And the tunica media, aka tunica musculosa, that's the smooth muscle layer that constricts or dilates. Okay. With sympathetic input, you have constriction and blood pressure can increase, along with hormones like norepinephrine, epinephrine. When the sympathetics don't engage and you're more parasympathetically engaged, which is more of a relaxed position, the smooth muscle relaxes. It also relaxes due to something the red blood cells can release, which is nitric oxide. Nitric oxide is stimulating the smooth muscle to tell it to dilate, and then blood pressure drops. Okay. So that's the tunica media or the musculosa, and then we have the artery here. And you have the external layer. The external layer is the tunica externa, and the tunica externa, aka tunica adventitia. Those are interchangeable terms. Okay, when we look here at the vein has valves right here. This is a valve. This is a valve. Notice arteries do not have veins, only arteries have veins. So once you see the vein, once you see the valve, valves are only found in veins, not in arteries. Okay, this is a dissected portion. Okay, let's look at the next slide. So now we're taking the model and we're rotating it around. I mean, from this view, we could see the artery, we could see the endothelial here, the tunica interna, we could see the smooth muscle layer, the tunica media or musculosa, and then we have the tunica adventitia, aka tunica externa. This is much more elastic. And then from the outer portion, when we turn it around, we could see some skeletal muscle here. We see some skeletal muscle here. And when skeletal muscles contract, they do help pump venous return of blood back to the brain or back up to the heart. Okay, so this is skeletal muscle. Here is a venous vein. This one is opened, and the one down here is closed. This around it, this is just fat or adipose. And the whole idea of carrying blood is one of the main functions is we want to bring oxygenated blood to the mitochondria of the other cells and tissues. So when the heart, when the all the blood goes to the right atrium, which is deoxygenated, and it goes down to the right ventricle, the right ventricle contracts inferior to superior, pumping the blood up into the pulmonary trunk, from the pulmonary trunk into the pulmonary arteries. The pulmonary arteries, which we see right here, the pulmonary arteries are going to bring the deoxygenated blood right down into the pulmonary arteriole. Now there's the exchange, right? There's the exchange because we have capillaries in here, and it's going to drive oxygen now into the pulmonary venule. From the pulmonary venule, it's going to pump it into the pulmonary vein, and the pulmonary veins from both the right and left lungs drive themselves into the left atrium. Veins bring blood into the heart. Arteries carry blood away from the heart. But when we think of arteries, most of the time, arteries are red and veins are blue. But when we look here at the level of the lung and the level of the heart, it's flip-flopped. If we look at the artery here, the pulmonary artery, the pulmonary arteriole, those are blue. If we look at the pulmonary venule and the pulmonary vein, those are red. Why are they red? Why is this vein red? It is carrying oxygenated blood back into the left atrium. Then from the left atrium, it goes down through the mitral valve into the left ventricle. The left ventricle contracts inferior to superior. When the blood pushes up, it shuts the mitral valve and opens up the aortic semilunar. When the aortic semilunar opens up, it goes up the ascending aorta, the arch of the aorta, and up through those main branches off the arch of the aorta that go up to the head, neck, and upper extremity. Then it goes down the descending aorta, thoracic aorta, abdominal aorta. Abdominal aorta has many branches that feeds the abdominal organs, oxygenated blood. And then it goes to the common iliacs, which will branch off into both lower extremities. Okay. And here's just another view from the opposite side, showing the pulmonary. Up here is the pulmonary artery. Here's the pulmonary arterial. And then on the red side, we can see the pulmonary venule and the pulmonary vein. Okay, so you have pulmonary artery, pulmonary arterial, pulmonary venule, pulmonary vein. And where they meet and communicate and do their exchange is at the alveolar capillaries, which are near the deep in the alveolar sac. Okay, so down in here, these are the alveolar alveoli, right, which is plural, or alveolus, which is singular. Here is the terminal bronchiole. This is what's bringing the oxygen in. Here's your pulmonary vein and pulmonary artery, and that's your bird's eye view looking straight down. Okay, here's your terminal bronchus. Okay, when we come back, we'll take a look at some of the blood vessels.